Good morning. My name is Beatriz Bolanos, and I am an epidemiologist at the Garcia Newgate Memorial Hospital. My team and I are here to inform the public of a new disease found amongst the patients. After detection of disease, we have taken these actions to ensure the safety of those in the hospital. We have quarantined those who are positive to prevent further spread and have required staff to utilize masks, gloves, and other equipment to protect themselves until we find a treatment for the disease. We enforce the washing of hands and the sterilization of every tool before use. Our main goal is to prevent cross-contamination as much as possible in order to pinpoint what this new disease is. My team and I have made progress on this case and are here to share our findings.
Good morning, my name is Mabel Perez Medina, an epidemiologist on the Garcia Newgate Memorial Hospital outbreak case. And today I'm here to discuss to you how we got the case defense team and how they got it figured out to be handled. In the hospital, when dealing with an outbreak like this, it does go through many steps or procedures. After the disease defense team, also known as the DDT, gets the call from the healthcare facility, they are able to identify and use information for this disease. From this, DDT will later be able to go on site and gather info using sources, including patient interviews and testing to get a better idea of the type of outbreak they are dealing with. They did this, and then the info is very important because it helps track the disease and helps with the prevention methods to deal with an outbreak. They can create prevention methods and identify risk factors associated, as well as what may be associated if you have any underlying conditions. After creating these, they can later check if it works, and using this they can make any revisions, and in the end, they all use this to help control and deal with this outbreak.
Hi, my name is David Torres, and I'm one of the epidemiologists on the Garcia Newgate Memorial Hospital outbreak case. Today, I'll be discussing what caused the virus and how it was spread. The disease was caused through the saline bags. The saline bags were most likely contaminated with a bacteria from outside with an unknown origin. All of the patients in the hospital had one common similarity between them, as they stayed in the hospital day in and day out, and it was the saline bags. All of the patients needed the IVs. Therefore, as the only common factor between all the patients, the IVs and the saline bags were the dangerous factor in the formula.
Hello and good morning. My name is Abigail Reyes, and I'm an epidemiologist working on the Garcia Newgate Memorial Hospital case. And I'll be telling you how my team and I figured out the bacteria that's infecting our patients. In order to figure out what was the identity of the pathogen, we first identified the category of infectious agent. Then we gathered patient samples and used them to grow the bacterium in order to observe their gross morphology. We grew the bacteria into isolated individual colonies by streaking them onto an agar plate, then incubating them upside down in an incubator at 30 degrees Celsius for 24 hours. This process is known as aseptic technique. Using this technique, we can observe the color, shape, elevation, edges, and size that may be different. We then use the gram staining process to find the cell morphology, arrangement, and gram stain result. Finally, we compared the results from the gram staining to bacterial genus and species to conclude that it's Stenotrophomonas maltophilia.
I'm Erica Hernandez, another scientist on the GNMH case. I was closely involved with the research that led towards the identification of the infection, so now I can tell you more about it. After conducting the test, we were able to identify the disease as Stenotrophomonas maltophilia. Its colony on the agar plate was large, yellow, circular, with an entire margin and convex elevation. From the gram staining process, we identified it as a single-cell bacillus with a gram-negative stain. With these results, we were able to further examine S. maltophilia and find out its symptoms. These included fever, shortness of breath, mucus, and chest pain, which were all symptoms shared by the patients. The way to treat S. maltophilia is through an antibiotic called SXT, used to treat or prevent infections.